We need to start with a question that it sounds almost childishly simple, but it's actually one of the deepest puzzles in the universe.
It's at the puzzle.
How do you get from a rock to a person?
Hmm.
Because if you actually look at the ingredients list for a human being or a tree or anything alive, it's shockingly boring. It's carbon, hydrogen, oxygen,
It's
nitrogen.
the same stuff you'd find in a cloud of interstellar dust. Literally stardust.
Right. And if I took those ingredients and put them in a giant pot and just stirred for a billion years, I'd end up with a mess. I wouldn't have something that can think and feel and, listen to a deep dive like this.
And that gap, that transformation,
Yeah.
that's the entire field of biochemistry. It's the bridge. On one side, you have chemistry, which is all about fixed laws, mathematical rules.
Predictable.
Very predictable. And on the other side, you have biology, which is messy, adaptive, and, well, alive. Biochemistry is the study of how the rules of the first side create the beautiful, chaotic complexity of the second.
So today, we're doing a deep dive into the absolute foundations of this field. We're not jumping into, a specific pathway or a new drug just yet. We are looking at the source code, the operating system of life itself.
And to guide us, we've pulled two of the giants. These are essentially the foundational texts for any medical student.
We've got Leninger's Principles of Biochemistry and Harper's Illustrated Biochemistry.
The Old and New Testaments, you could say.
I was thinking that too. And they really aren't just two different versions of the same book, are they? Reading them side by side for this, they felt like they have completely different personalities.
Oh, completely. You can think of Leninger as the architect. It's obsessed with the logic of life, the systems thinking. It wants you to understand the physics, the thermodynamics, the structural beauty of why a protein folds exactly the way it does.
It treats the body like a perfect, elegant machine.
Exactly. Whereas Harper, to me, feels like the ER doctor who just burst through the doors covered in,
Yeah. No time for elegance.
No time. Harper is all about the clinical reality. It's less concerned with the abstract beauty and much more concerned with what happens when that machine breaks. It says, okay, here's the pathway, but here's the mutation that shatters it, here's the disease it causes, and here's how we try to fix it.
So our mission today is to synthesize those two perspectives. We want the architectural why from Leninger combined with the life or death so what from Harper. We're going to look at the origins of the field, the physics of the cell, the geometry of carbon, and the energy laws that, literally keep us from falling apart.
And we should be really clear here for anyone listening, especially students. This is a synthesis. It's a guide. We're not just reading the textbook to you.
Right.
You still need to read the books. Our job is to build the mental scaffolding, to give you the Medlock Holmes mindset, as you put it, so that when you read the chapters, you see the patterns. You understand why it matters.
I do like Medlock Holmes. Okay, let's start at the crime scene. Or the origin story, I guess. You mentioned we're made of stardust. Leninger actually opens with this very grand cosmic perspective.
It does. It zooms all the way out. It reminds us that the universe started as this incredibly hot, dense soup of energy and particles.
The Big Bang.
Right. And as it cooled over billions of years, you get simple elements, hydrogen, helium. Then those elements collapse into stars. And inside the nuclear furnaces of those stars, the heavier elements get forged.
Carbon, nitrogen, oxygen.
The stuff of life.
And when those stars die,
They explode, supernovae. They scatter that precious dust across the cosmos. That dust eventually formed planets like Earth. And on this planet, that same dust somehow organized itself into bacteria, then plants, then us.
It creates this incredible paradox, though, because if I take a sample of my own liver and I put it in a mass spectrometer, the carbon atoms inside me are identical to the carbon atoms in a lump of coal. They obey the exact same laws of physics. There's no special life gravity or biological electricity. It's all the same rulebook.
And that, right there, was the single biggest mental hurdle for early scientists to get over. Because for centuries, for millennia, everyone was convinced there was something special.
The vital force.
Nihilism, yeah. It was the dominant theory until, really, the end of the 19th century. The idea was that biological reactions, things like fermentation or digestion, they could only happen inside a living cell.
Why?
Because life was thought to have this unique, intangible spirit, a vital force.
Yeah.
You couldn't make urea in a beaker because you couldn't bottle the ghost in the machine.
So there's this hard wall. Chemistry was for non-living things, rocks and minerals. And biology was for, for things with a soul.
Essentially, yes.
Hmm.
A hard distinction between the living and the non-living. But Harper tells the story of how that wall was just smashed to pieces. And it wasn't by some grand philosophical insight.
It was an accident, wasn't it?
It was an accident by two brothers in 1899, Everett and Hans Buchner.
The yeast guys.
The yeast guys. And you have to understand, they weren't trying to discover the meaning of life. They were trying to make a medicine, a cell-free extract of yeast for therapeutic use.
So they took yeast cells and they mixed them with sand to act as an abrasive. They put it all in a mortar and pestle and just ground it until every single cell was crushed, obliterated.
Then what?
Then they put this paste into a canvas bag and squeezed it with a hydraulic press.
Wow, that's brutal.
It's very brutal. And what dripped out was this reddish, cell-free juice. They called it zymase. Now, according to all the science of the time, this juice was dead.
Right. No cells, no vital force. It should just rot.
Correct. So to preserve it, they decided to add a bunch of sugar, like making jam, sucrose.
Preservative. Makes sense.
But then the impossible happened. The dead juice started to bubble.
Bubbles.
Bubbles vigorously.
Carbon dioxide.
And alcohol. The juice was fermenting. It was running a complex, multi-step metabolic pathway, converting sugar to ethanol and CO2 without a single living cell present.
That had to be a complete shock.
It was earth-shattering. It proved, in one experiment, that life wasn't some mystical spirit. It was a series of chemical reactions, catalyzed by enzymes. You could take the machinery out of the factory, put it on the floor, give it fuel, and it would still run.
That's the birth of biochemistry.
That's the moment, and it completely reframes what we are. We're not magical. We're just incredibly complex, wet, squishy machines, but machines that run on chemical logic.
And once you accept that, you can finally define health in a rigorous way. Because if we're machines, then health is just the machine running correctly. Harper has this definition that I think is just brilliant.
I've got it right here. It says, health is a situation where thousands of intra- and extracellular reactions proceed at rates commensurate with survival.
The key word there is rates.
Why rates? Why not just, the reactions are happening?
Because biology is all about timing and quantity. It's a dynamic flow. You might have the right enzyme, you might have the fuel, but if the reaction runs 50% too slow,
you don't make enough ATP to keep your heart beating.
Right. Or if it runs 50% too fast, you might burn through all your glucose and slip into a coma.
Yeah.
It's not just about on or off. It's about the speed, the rate.
So disease isn't always a broken part. It can just be a timing error, a regulation problem.
That's what Harper argues. Almost all disease is, at its root, a disturbance in biochemistry. Think about it. Dehydration is an electrolyte imbalance messing with nerve conduction. Diabetes is a failure in the signaling pathways for glucose. It's all chemistry.
And this creates a two-way street, right? The book mentions that biochemistry explains disease.
But disease also teaches us about biochemistry.
How so?
Historically, that's often how we figured out how the normal machine works, by studying the broken ones. These rare, tragic genetic diseases become windows into normal function.
You're talking about things like sickle cell anemia?
That's the classic example. Linus Pauling famously called it the first molecular disease. By studying these sick patients, scientists were able to pinpoint a single tiny error.
In the DNA.
A single point mutation. One amino acid in the entire hemoglobin protein chain, a glutamate, was swapped for a valine.
Just one switch out of hundreds.
Just one. But here's the chemistry. Glutamate is hydrophilic. It loves water. Valine is hydrophobic. It hates water. So you swapped a water-loving component for a water-hating one on the surface of the protein.
And what does that do?
When oxygen levels are low, that hydrophobic valine patch causes the hemoglobin molecules to stick together, to polymerize. They form these long, rigid fibers that distort the red blood cell.
Into the sickle shape.
Into the sickle shape.
Ah.
And that's the disease. So you see the chain of command. One tiny typo in the genetic code leads to one change in a chemical property, which changes the protein structure, which changes the cell's shape, which changes the blood's flow, which causes the organ damage.
Molecular to cellular to physiological. Wow.
It's the whole hierarchy in one example.
Okay, so let's zoom in on that stage. The cell. We've established that reactions drive life, but they don't happen in some abstract void. They happen inside the cell.
The fundamental unit.
Now, Leninger spends a lot of time on this, and it really tries to dismantle a common misconception. I think most of us, when we picture a cell, we see that diagram from a high school textbook.
The big empty balloon.
Yeah. A big blue circle, a nucleus in the middle, a couple of mitochondria scattered around, and just A lot of empty, watery space.
The bag of water model. And it's profoundly misleading.
It makes you think that molecules are just sort of floating around randomly until they bump into each other.
Right. But Leininger paints a very different picture. The cytosol, the fluid inside the cell, is not water. It's a dense, viscous, highly concentrated gel.
A gel.
Think less like tap water and more like egg whites or Jell-O. It is packed to the brim with proteins, ribosomes, metabolites, cytoskeletal fibers. The molecular crowding is immense.
So what does that crowding do to the chemical reactions? How does it affect them?
It fundamentally changes the physics of movement. In a dilute solution in a test tube, what we call in vitro diffusion is pretty fast. Molecules can zip around. But inside a cell in vivo diffusion is much, much slower.
It's like trying to run through a packed concert versus an empty field.
That's a perfect analogy. It's a random walk through a very crowded dance floor. A molecule can't just expect to float across the cell to find its partner enzyme. It would take too long.
So how does the cell solve that?
Organization, compartments and complexes. Enzymes that work together in a pathway are often physically stuck together. They form these metabolines, these molecular assembly lines.
Like a bucket brigade.
Exactly like a bucket brigade. The product of reaction A is immediately handed off to enzyme B, which uses it as fuel for reaction C. The intermediate molecule never even has to enter the crowded dance floor. It just gets passed along.
Efficiency born from necessity. Okay, so there's another physical constraint the books bring up, and it answers a question I've had since I was a kid.
Let's hear it.
Why aren't there single-celled organisms the size of a dog? Or a basketball? You know, the classic movie monster, the blob.
Ah, the blob scenario.
Yeah, why is life made of trillions of tiny cells instead of just one enormous one?
It comes down to a really ruthless, inescapable law of mathematics, the surface-to-volume ratio.
Okay, walk us through the math on that.
Imagine a perfectly spherical cell. As it gets bigger, its volume, which represents its metabolic needs, its hunger increases by the cube of its radius, r cubed.
So it gets hungry fast. A bigger factory needs more power.
Right. But its surface area, the cell membrane, which is its mouth, the only way to get fuel in and waste out, that only increases by the square of the radius, r squared.
Oh, I see where this is going. The hunger grows way faster than the mouth does.
Precisely. R cubed always outpaces r squared. If a cell gets too big, its central volume is too far from the surface. It essentially suffocates and starves. Oxygen and nutrients simply cannot diffuse from the surface to the center fast enough to meet the metabolic demand.
So there's a hard physical limit on how big a single unit of life can be.
There is, unless you cheat the math.
And life always finds a way to cheat.
Always. Leininger points this out. Look at cells that need a large surface area, like the cells lining your intestine that absorb nutrients. They're covered in microvilli.
These tiny finger-like folds.
Exactly. Folding massively increases the surface area, the r squared, without dramatically increasing the volume, the r cubed. It's an evolutionary hack to beat the geometric constraints. It allows us to build a large, complex organism while keeping all our cells within a safe distance of their supply lines.
Okay, so we've built the stage. A crowded, gel-filled cell with strict size limits. Now let's talk about the actors on that stage, the atoms themselves.
The cast of characters is surprisingly small.
It really is. When you break it down, 99% of the mass of a cell is just 4 elements: hydrogen, oxygen, nitrogen, and carbon.
HONC. That's pretty much it.
So why those 4? Out of all the elements on the periodic table, why did life choose them? Why not, I don't know, lead and gold and argon?
It's all about bond strength and stability. Leininger explains that to build reliable machinery, you need strong, stable connections, covalent bonds. And it turns out that lighter elements form stronger covalent bonds.
So HON and C are just the lightest elements that can form the right number of bonds. 1, 2, 3, and 4, respectively.
That's the core reason. They provide the strongest, lightest building materials.
Yeah.
But among those 4, the real superstar, the most valuable player, is carbon.
We hear the phrase carbon-based life all the time, but let's break that down. Why is it so special? Silicon is right below carbon on the periodic table. It also has 4 valence electrons. It can form 4 bonds. Why aren't we silicon-based life? Why aren't we having this discussion as, rock monsters?
It's a great sci-fi question, but biochemically it's a non-starter. There are a couple of huge reasons. First is bond energy. Carbon single bonds are remarkably stable. They have an energy of about 350 kilojoules per mole. Strong enough to build with, but not so strong that our enzymes can't break them down for energy. Silicon bonds are significantly weaker.
So our skeletons would be more brittle.
Much more brittle, especially in the presence of water. But the bigger problem, the absolute deal breaker, is waste management.
Waste management.
Think about what happens when you metabolize sugar. You're breaking down carbon compounds. What's the waste product?
Carbon dioxide, CO2.
Which is a gas at body temperature. It's soluble, you can transport it in your blood, and you can exhale it. It's a perfect disposable waste product.
Okay. What happens with silicon?
When silicon fully oxidizes, you get silicon dioxide, SiO2.
Which is sand.
Quartz, a solid crystal, a rock. Imagine if every time you burned a molecule of fuel, you had to exhale a tiny grain of sand.
That would be a problem. Medically, that would be a huge problem.
It'd be impossible. Your lungs would turn to stone. Carbon's chemistry allows for the flow and circulation of matter. Silicon's chemistry leads to geology. It just sits there. Plus, carbon is just so versatile. It can form single, double, triple bonds. It can make long chains, branch chains, complex rings. It's like biological Lego.
But Leninger takes this a step further. It says it's not just about which atoms are connected. It's about how they are arranged in three-dimensional space.
And this is where we get into stereochemistry. It's a topic that sounds really abstract and academic until you realize it's the difference between a medicine and a poison.
This is the concept of chirality, right? Handedness.
Exactly. The easiest way to get it is to just look at your own two hands. Your left hand and your right hand have the same components: a thumb, 4 fingers, a palm. But they are mirror images.
And you can't superimpose them. If I try to put my left hand flat on top of my right hand, the thumbs are pointing in opposite directions.
They're non-superimposable mirror images. You can't fit a left-handed glove on your right hand. It just doesn't work.
And molecules can be like that?
Carbon molecules can. If a carbon atom is bonded to 4 different groups, that carbon becomes a chiral center. It can exist in 2 forms, a left-handed version and a right-handed version. They're called enantiomers.
And here's the part that is just so bizarre to me. Life is incredibly ridiculously picky about this.
Oh, it's an absolute tyrant about it. The 20 common amino acids that make up every single protein in your body, they are all, with one tiny exception, L-isomers. Left-handed.
And sugars.
Sugars, like the glucose that's powering your brain right now, are almost exclusively D-isomers. Right-handed.
Why? Why this uniformity?
We don't know for sure why those specific hands were chosen at the dawn of life. It might have been a cosmic accident. But once the choice was made, it had to be consistent. Think about it. Enzymes are proteins, right? They're big, complex 3D structures with very specific pockets in them called active sites.
The gloves?
They're the gloves. An enzyme is like a very specific left-handed glove. It's built to perfectly fit a left-handed amino acid. If a right-handed amino acid comes along, it just won't fit in the pocket. It can't bind. No reaction.
The whole system has to use the same convention or nothing fits together.
The machinery is stereospecific. And this has huge implications for medicine. Leninger gives a great clinical example, the antidepressant citalopram. Right. It's a very common SSRI. When you synthesize citalopram in the lab, you get what's called a racemic mixture, a 50-50 mix of the left-handed and the right-handed molecules.
The S isomer and the R isomer?
Exactly. On paper, they look identical. Same atoms, same bonds. But in the three-dimensional reality of your brain, only one of them actually works. The S isomer is the one that fits into the serotonin transporter and produces the antidepressant effect.
So what does the other one do, the R isomer?
Nothing useful. It's just dead weight. It doesn't fit the lock. In some drugs, the wrong isomer can even be harmful or cause side effects.
Which is why drug companies eventually figured this out.
They did. This is why you can also get a prescription for escitalopram, which is sold as Lexapro. The S stands for the S isomer.
So it's the same drug, just purified.
It's literally just the active half. They filtered out all the useless R isomer. This means you can get the same therapeutic effect with half the dose, which often means fewer side effects.
It's amazing that a tiny flip in geometry, something you can't even see, is the difference between an effective medicine and a useless chemical.
And we owe the discovery of this whole concept to one person, Louis Pasteur.
The same guy who invented pasteurization for milk?
The very same. Long before he was working on food safety, as a young man in 1843, he was studying tartaric acid crystals that were precipitating out of wine casks.
Okay.
He put them under a microscope, and he noticed something no one had ever noticed before. There were 2 slightly different types of crystals. They were mirror images of each other.
And he separated them.
He sat there with a pair of tweezers and by hand painstakingly separated the pile of left-handed crystals from the pile of right-handed crystals.
That must have taken forever.
I can't even imagine. But then he did the brilliant part. He dissolved each pile in water separately and shone a beam of polarized light through each solution. The solution with the left-handed crystals rotated the light to the left. The solution with the right-handed crystals rotated it to the right. That was the moment. The discovery of molecular asymmetry. It underpins all of modern pharmacology and biochemistry.
So we have the stage, the cell, we have the materials carbon, and we have the precise 3D shaped stereochemistry. But none of this matters without power. A machine doesn't run if it's not plugged in.
Which brings us to thermodynamics, the laws of energy.
And Leninger makes a critical point right at the start of this section. It says that living organisms are open systems.
That's a crucial distinction. It means we are not sealed jars. We are constantly exchanging both matter and energy with our surroundings.
We eat, we breathe, we excrete.
Right. We take in high-energy, complex matter food, and we release low-energy, simple waste carbon dioxide and heat. We are a conduit for the flow of energy.
And we have to do this to fight against a fundamental law of the universe. The second law of thermodynamics.
Entropy, the relentless tendency of the universe to move towards disorder, towards randomness, towards chaos.
If you leave a building alone for 1,000 years, it becomes a pile of rubble. It never spontaneously organizes itself from rubble back into a building.
Correct. But life, life does the opposite. We take disordered, simple molecules from our food, and we build highly ordered, complex structures. We build proteins, DNA, cells. We create order.
We are fighting against the current.
We are a small, local island of low entropy in a vast, chaotic ocean of high entropy. But maintaining that island, fighting that current, comes at a cost. We have to constantly consume energy to pay for the order we create.
And this leads to what might be the single most important concept in this entire foundation: the difference between equilibrium and steady state.
Yes. This is where our everyday intuition can lead us astray. In normal language, equilibrium sounds like a good thing. Peace, balance, harmony.
Sounds nice and relaxing.
In biochemistry, equilibrium is death. Period.
Unpack that. Why is it so final?
Because thermodynamic equilibrium means there is no net flow of energy or matter. It means everything is evened out. The battery is dead. The concentration of ions inside and outside your cells is the same. The temperature of your body is the same as the room.
So if your chemical gradients reach equilibrium, your nerves can't fire. If you reach thermal equilibrium, you're dead.
It is the ultimate state of disorder and inaction. So the entire project of life is to maintain a state that is as far from equilibrium as possible.
And that state is called a dynamic steady state.
Yes. Leninger uses a great bathtub analogy. Imagine a bathtub, but the drain is wide open.
Okay, so water will just run out.
But the tap is also running at full blast. And the rate at which water is flowing in from the tap is exactly equal to the rate at which it's flowing out the drain.
So the water level in the tub stays the same?
The level stays constant. From a distance, it looks static. It looks like a system at equilibrium. But it is violently dynamic. A huge amount of water and energy is flowing through the system to maintain that steady level. The actual water molecules in the tub are being replaced every few seconds.
That's us.
That is you. The amount of glucose in your blood stays in a narrow, steady range. But it's not the same glucose molecules. You are constantly using glucose and constantly releasing more from your liver to maintain that steady state. The rate of synthesis equals the rate of breakdown.
And keeping that tap running, maintaining that flow, that's what costs all the energy.
That is the energy cost of being alive. When you feel tired at the end of the day, that's the feeling of having spent 12 or 16 hours burning fuel just to keep your system away from the drain of equilibrium.
So how do we actually pay that energy bill? You know, we don't plug into a wall socket. How does the cell do it?
It uses a strategy called chemical coupling. This is sort of the economic model of the cell.
Okay, coupling. What does that mean?
Many of the reactions life needs to run, like building a muscle protein or synthesizing DNA, are endergonic. They require an input of energy. They're thermodynamically unfavorable. They are an uphill battle against entropy.
It won't happen on their own.
Never. So the cell has to be clever. It takes that energetically unfavorable uphill reaction and it couples it or links it to a separate reaction that is massively favorable and downhill, a reaction that releases a huge amount of energy.
And the downhill reaction is the hydrolysis of ATP.
Adenosine triphosphate, the universal energy currency of the cell.
What makes ATP so special? Why is it the currency?
It's all about its structure. ATP has a chain of 3 phosphate groups attached to it. Each of those phosphate groups is negatively charged.
And negative charges repel each other.
Viciously. So you have three negative charges be forced to sit right next to each other in this molecule. It's like trying to compress a powerful spring or hold the north poles of three strong magnets together. That phosphoanhydride bond is under immense strain,
So it's full of potential energy.
a huge amount. And when an enzyme cuts that final phosphate group off hydrolyzing ATP to ADP and a free phosphate, that spring uncoils. The phosphates fly apart because of that electrostatic repulsion.
And that snap releases a burst of energy.
It releases a packet of free energy, about negative 30.5 kilojoules per mole under standard conditions. The cell then cleverly harnesses the energy from that ATP explosion to pay for and drive the uphill reaction it needed to run.
So it's a direct transaction. Burn one ATP to add one amino acid to a protein chain.
It's exactly that. A transaction that happens billions upon billions of times per second in every one of your cells. If you were to stop all ATP synthesis in your body, you would run out of energy and reach equilibrium and die in under a minute.
That is incredibly high stakes.
Wow.
Okay, so we've covered the history, the cellular stage, the chemistry of carbon, and the laws of energy. Before we wrap up, I want to touch on where this is all going, the future.
The omics revolution.
Right. Harper makes the point that the completion of the Human Genome Project back in 2003 really changed the game. But we're now moving way beyond just looking at genes.
We are. For most of the 20th century, biochemistry was very reductionist. You studied one enzyme at a time, one pathway. I'm the world expert on hexokinase.
Now we study the entire system at once.
We had genomics, which is the complete set of genes, transcriptomics, which is all the RNA messages being sent from those genes, proteomics, all the proteins being built, and now the really exciting frontier is metabolomics.
What does metabolomics tell you that the others don't?
Okay, think of it this way. Your genome is the library of blueprints. It's the master plan. It tells you what your body could potentially build.
But it doesn't tell you what's actually being built right now.
Precisely. The metabolome is the real-time snapshot of what's actually happening. It's the measurement of all the small molecules, the glucose, the lactate, the amino acids, the hormones that are actually circulating in your blood and tissues right now. It's the functional output of your genes interacting with your diet, your environment, your stress levels.
So if the genome is the architectural blueprint of the city, the metabolome is the live traffic report.
That's a perfect analogy. And for developing personalized medicine, the traffic report is often what you really need. Harper suggests that in the future, we won't just treat type 2 diabetes. We'll get your personal metabolomic profile, your specific traffic report, and design a therapy to fix your specific traffic jam.
It brings us right back to that definition of health from the beginning. It's about monitoring the rates of thousands of reactions to ensure survival.
We're finally building the tools to see the whole machine running in real time.
This has been a huge journey today. I mean, we've gone from the Buckner brothers grinding up yeast with sand in 1899
to the physics of molecular crowding in the cytosol and the mathematical limits on cell size.
We talked about the unique versatility of carbon and the life or death importance of 3D shape in stereochemistry.
And we ended with the constant thermodynamic battle every organism wages just to maintain that dynamic steady state far from equilibrium.
I want to leave our listeners with one final thought. It's from a section in Leninger that I just found so profound. It connects all of this biochemistry to Shakespeare.
Ah, yes. The entropy of information.
Right. The text quotes a line from Julius Caesar. There is a tide in the affairs of men. It's a beautiful, complex, information-rich sentence.
And from a thermodynamic perspective, it is a very low entropy state. It's highly ordered. The letters are arranged in a very specific, non-random sequence to convey meaning.
But if you took all the letters in that sentence, cut them out individually, and threw them in a box and shook it, you would just have a jumble of letters, confetti.
High entropy, maximum disorder, and zero information.
The component parts are identical. The mass is the same. The elements are the same. But the meaning, the function, is completely gone.
And that is the physical difference between a living organism and a corpse.
You listening to this right now are that Shakespeare quote. You are a highly ordered, low-entropy, information-rich collection of stardust. And the entire universe, the second law of thermodynamics, wants you to be confetti. It wants to scramble your letters back into alphabet soup.
But you are fighting back. Every single ATP molecule your body burns, every enzyme that catalyzes a reaction, every breath you take is an act of defiance against that universal pull toward chaos. You are spending energy constantly just to keep your letters in the right order.
So the next time you feel tired, when you feel physically and mentally exhausted at the end of a long day,
Yeah.
just remember what that feeling is. It's the tangible metabolic cost of being a complex, meaningful story in a universe that prefers random noise.
It's the price you pay for your steady state.
Keep fighting the entropy. Thanks for listening to The Deep Dive.
Stay curious.